This is the second video about lipid metabolism to help you get ready for your final. This video talks about beta oxidation and ketogenesis. Beta oxidation is a great way to generate energy from stored fat when there's no glucose readily available in a cell. Here's the big picture. Essentially, you start with one activated fatty acid, and each time you put it through one round of beta oxidation, you create one molecule of acetyl-CoA by breaking off the first two carbons along with the attached coenzyme group. That same fatty acid can be put through continuous rounds of beta oxidation until all of the carbons are gone. Beta oxidation occurs in 4 steps. The first step is an oxidation-reduction reaction in which the two carbons that are closest to the carbonyl carbon are oxidized as FAD is reduced to FADH2. The second step is a hydration reaction that adds water back into the bond. The third step is another oxidation-reduction reaction in which NAD+ is reduced to NADH and hydrogen ion as the fatty acid molecule is once again oxidized, forming a new carbonyl carbon. In the fourth and final step of beta oxidation, a new molecule of coenzyme A bonds to the new carbonyl carbon, releasing a molecule of acetyl-CoA. Let's review that really quick. The first step is an oxidation with FADH that generates one molecule of FADH2. The second reaction is a hydration reaction. The third reaction is an oxidation with NAD+ that generates one molecule of NADH and hydrogen. And the final reaction is the bonding of a new molecule of coenzyme A that releases a full molecule of acetyl-CoA. The next thing to think about is what happens to all of that acetyl-CoA. Generally, acetyl-CoA goes directly to the Krebs cycle. However, it's possible that it can meet a different fate in the liver. The liver packages acetyl-CoA into what are called ketone bodies. Ketone bodies are water soluble, so they can be transported to the rest of the body's tissues through the blood. The body's cells will take up the ketone bodies, convert them back into acetyl-CoA, so that the acetyl-CoA can enter the Krebs cycle and participate in electron transport. Ketogenesis happens in 4 steps. The first reaction is a condensation reaction that combines 2 molecules of acetyl-CoA into one molecule of acetoacetyl-CoA. The second reaction is another condensation reaction that adds another molecule of acetyl-CoA to form 3-hydroxy-3-methylglutaryl-CoA. In the third reaction, this 6-carbon molecule is then degraded to acetoacetate by removing a molecule of acetyl-CoA. One of 2 things can happen to acetoacetate. It can either undergo an enzyme-catalyzed oxidation-reduction reaction in which NADH and hydrogen ion are oxidized as acetoacetate is reduced to 3-hydroxybutyrate, or it can undergo non-enzymatic decarboxylation, which produces one molecule of CO2 and one molecule of acetone. Acetoacetate and 3-hydroxybutyrate are useful to the body, whereas acetone is really just a waste product. So what happens when 3-hydroxybutyrate and acetoacetate make it back into the body cells? They're converted back into acetyl-CoA and pushed back into the TCA cycle. 3-hydroxybutyrate can be oxidized back into acetoacetate, in the process reducing an NAD+ to an NADH and hydrogen ion. At this point, succinyl-CoA, which you should recognize from the Krebs cycle, can donate its coenzyme A group to produce another acetoacetyl-CoA, itself becoming succinate. Another molecule of coenzyme A binds to a carbonyl carbon, splitting acetoacetyl-CoA into 2 molecules of acetyl-CoA. This acetyl-CoA can then enter the TCA cycle, and nobody is going to starve to death. That's it for this video. The next video is going to cover fatty acid synthesis.